Alright, here we're talking a little bit more about the nucleus and ribosomes. We've talked some about this. The lab certainly had some of this in it. But of course, the nucleus contains most of the DNA. The ribosomes are used to read the DNA and put together a protein. Now, the nucleus contains most of the cell's genes. Alright, there is some DNA in a mitochondria or chloroplast. That was explained in a different video. But the nuclear envelope is basically the membrane, double membrane, that encloses the DNA and essentially forms the nucleus. And this is kind of what the nucleus looks like. Notice that it is surrounded by the ER. This here is the rough ER, rough meaning that it has ribosomes stuck to it. Here is an electron microscope picture of what the nucleus would look like. And there are, you can actually see these little pores in the nucleus and in the ER to let the RNAs out of the nucleus, and you can see that here in the electron microscope. So these little pores allow the mRNA to leave and go into the cytoplasm so that they can be read by a ribosome. This is actually what the DNA is all wrapped up here. That's showing the nucleus. We call it the chromatin when you look at it closely. Here's just another picture of the nucleus, all blown up. Again, the same picture that we saw before. So recognize the pores are there to let the mRNA out. You need to understand that. Just recognize that you have to let the RNA out. That's kind of the idea, and that there are pores in the nucleus that have a specific task to do that. DNA is organized in chromosomes. We talked about in the last time that sometimes you'll say that there's a bacterial chromosome, and that is slang because they have circular DNA, and chromosomes must be linear. So that is the correct thing is to call them a genophore, or their nucleoids, and all kinds of things that they might call the bacterial DNA. But here in a eukaryote, we call them chromosomes. And whenever you mix DNA and the proteins that they wrap around, we call it chromatin. You need to know that term. And you need the nucleolus, which is kind of a special area inside the nucleus where certain parts of the chromosomes kind of hang out, and they make ribosomes. All right, so you need to know that the nucleolus is a special place. Lots of activity there. So ribosomes themselves are little machines, chemical machines that can read. They're like a chemical computer. They read the DNA or the RNA, and we should say it correctly and say RNA, and they read those instructions for making proteins. And so a ribosome is like a little factory that reads. You send it some instructions and it builds you a protein, and those instructions come from the nucleus, where the DNA is copied into RNA. So you have all the instructions in the nucleus, but you only send out certain instructions out at a given time for the ribosomes to make certain proteins. So let's see. This is what a ribosome looks like. It has here. These are the two proteins separated. It's a computer model of what the of both proteins. It's make for sure. The ribosome is made of two. Is a set of actually some RNA and proteins are all a piece of gum that has been on the ground, and that's kind of like subunits look like. Really, it's a bunch of two subunits come together, and when they do, RNA, and they begin a protein. It's a little factory. Ribosomes can be There are free ribosomes that are just floating around. And if you have ribosomes stuck to the stands for endoplasmic reticulum. That is fine. We saw this before, and this topic outside of the book.